Hello everyone. Today we are going to wind up this chapter, and this chapter that is biotechnology, principle and processes. We are going to talk about the last session of this chapter. In last three classes we have talked about genetic engineering, bioprocess engineering, then we talked about the tools of recombinant DNA technology. Then in last class we have started the processes of this recombinant DNA technology, and today we are going to wind up that. So let's start. Disclaimer. So, what are going to be the learning objective of today's class? First, we will understand the key steps in recombinant DNA technology. The key step involves isolation, cutting, joining, transfer. Isolation of DNA we have studied in the last class, but we are going to touch this topic again today. Cutting, that is the using restriction endonucleases for cutting our DNA at the specific point. Then joining of the foreign DNA and vector DNA using DNA ligase, and then transferring of that recombinant DNA into host cell. Second learning objective will be that we will identify the essential enzymes and vectors used in this recombinant DNA technology. Then we will comprehend the selection and screening of the recombinants, and lastly analyze the applications in biotechnology. So this is our today's index. First we will cover the 9.3 session of NCERT, which is the processes of recombinant DNA technology, ISO, whose subsections are isolation of genetic material, that is DNA, cutting DNA and separation of those fragments, then amplification using PCR, that is polymerase chain reaction, that we're going to talk in detail, then ligation of DNA into a vector, then insertion of that recombinant DNA which has formed after the ligation of foreign DNA and vector into the host. Then last we will see that how we can obtain the foreign gene product and what are those downstreaming processes. Then we will give a gist of all those things that we have gonna talk about in this whole class in our summary session. Then there will be NCERT based questions, and there will be some interesting keynotes and some interesting questions that you guys gonna solve yourself. So first step, that is the isolation of genetic material, that is DNA, which we have talked in the last class, but today we are going to summarize it. First, what we have studied there, that first we prepare the sample. We lyse the cell using some detergents and enzymes to break the cell membranes and release DNA. In last class we have talked like how we use cellulase for plant, lysozyme for bacteria, and chitinase for chitin. Then we remove the protein and RNA using protease enzyme, that and as well as RNase enzyme. This RNase enzyme eliminates the RNA contamination. So like this we will be getting free of all those cell membranes, proteins and RNA, and we will get our pure DNA. So here is the sample preparation, how first we lyse the cell wall, then how this, we add those all those detergent and all those things, then how we add the chilled ethanol to precipitate our DNA, and then we elute our DNA. This is the steps of DNA isolation. So as I told, after all the removal of, after breaking the cell wall, cell membrane, and removing this RNA and protein, we add chilled ethanol or isopropanol, often with sodium acetate, and we precipitate out our DNA. So let's start. So in the last step, we add this chilled ethanol or isopropanol, often with sodium acetate, and precipitate out our DNA. This DNA, after this precipitated DNA is washed with ethanol to remove any impurities, and as we have studied in the last class, using spooling method we can swirl that DNA around that spool, or you can say glass rod, and take out our DNA. Now we got this pure DNA. Now what we will do, we will dissolve this pure DNA into a suitable buffer. For example, tris EDTA buffer. This tris EDTA buffer usually used to keep this DNA and RNA stable, and we use this buffer for DNA suspension, for suspending this pure DNA. Here are the steps that how we prepare the sample. First lysis, then washing and DNA elution, all those steps. Now, after isolating that DNA, after we got our pure DNA, now it's time to cut our DNA at the specific position so that we can get our desired gene. Okay, so how we can get our desired gene? From last few classes, we know that restriction enzymes, restriction endonucleases are used to cut the DNA at the specific position. Okay, so this purified DNA is incubated, is kept with the chosen restriction enzyme under optimal condition for that enzyme. So we will take our pure DNA, we will keep it with our chosen restriction enzyme at the condition where that enzyme can work. Then this restriction enzyme will cut the DNA at some specific sequences and create two ends, sticky ends or blunt ends. As I have already told about the sticky ends or blunt ends in previous classes, still I will I will tell you today again that sticky ends are the overhang stretches, because in this restriction enzymes not cut directly in the middle of both the strand. It cut between the same bases in the complementary strands, but it creates the overhang stretches. But in blunt end, they the restriction enzyme cut both the DNA strand at the same position. Common enzymes we use is ECOR, R1, BAM-H1 and HIN3. So here's the pictorial presentation of that. Here you can see that how restriction endonucleases had cut this DNA such a way that this sticky ends created, this overhang stretches has created, and now this overhang stretches is going to join together to form this recombinant DNA in this step. Okay, let's go. Okay, yeah, so now not only this DNA, this foreign DNA, the desired DNA we need to cut at specific position, but our vector DNA, which is going to hold this donor DNA, is also be cut with the help of restriction endonucleases. And as I have told in earlier class also, same restriction enzyme should be used for donor DNA as well as vector DNA. It enables the compatible ends for joining. It's essential for creating the recombinant DNA, because definitely if it won't going to cut the donor and vector DNA, how it is going to join, or how this recombinant DNA is going to make. This process is occur in lab condition, in a controlled lab conditions. It requires the knowledge of genetic engineering, and this is again the same pictorial representation, how the sticky ends generate and then they join together. Now we got our gene. Okay, like I need this foreign gene, and I have isolated my DNA, pure DNA. I have used restriction enzyme, I have got my DNA. Now before joining it to vector, I need my gene of interest to got amplify, to get more copies of it. And how we can do it? We can do it by using PCR, that is polymerase chain reaction. we will study. So now what is PCR, polymerase chain reaction? What is purpose? So its purpose, as I told, is to synthesize multiple copies, that is amplification of a specific gene or a DNA sequence in vitro. In vitro, you guys know that in what is in vitro? So in vitro is the experiments or procedures that are carried outside of a living organism in a laboratory in a control settings. So we can synthesize the multiple copies of a specific gene of our gene of interest in vitro outside the living beings in the laboratory. So here is the pictorial representation. This is a piece. This is how a thermal a PCR look likes, and these are the PCR components which we're gonna talk each by each in next coming slides. And this is the basic step of the PCRs that we also gonna know in detail. Let's go. So process. What is the process of amplification of of our gene of interest using PCR? So yeah, before like we need to amplify our gene of interest. So for that, what do we need? First of all, we need that DNA template, that DNA that need to be amplified. Second, we need primers. Now, what are primers? So primers, as it says, two set of chemically synthesized small oligonucleotides, means small structures of nucleotides that are complementary to the regions of the DNA flanking the target sequence. In short, primers are small chemically synthesized short strand nucleotides that are complementary to the region of the DNA that we wanted. Okay, you will get to understand it more in next if when I will tell you this in this picture form. So first we need the DNA template, the DNA that need to be amplified. Second, the primers, the set of small oligonucleotides that are complementary to the region of the DNA that we need to amplify. And third, DNA polymerase. So what is a DNA polymerase in general? So it is an enzyme that synthesizes DNA molecules by adding nucleotides to a growing DNA strand. In this, when we have a DNA template, primer will go and bind to its complementary region. Okay. But this DNA polymerase, what will do? It will go and start adding nucleotides to that growing DNA strands. To that primer has to extend now. So this DNA polymerase is going to add nucleotides to the growing DNA strands. Here in this PCR, there is a specific DNA polymerase used, which is Taq polymerase, which is isolated from a bacterium which is called Thermus aquaticus. What is its speciality? That it remains active at high temperature also. And why we need that it should activate in high temperature also? Because the steps we follow in PCR that we're gonna talk in next slides, these steps of PCR they are steps which need high temperature. That's the reason that we use this DNA polymerase, because it can withstand even high temperature. It is very thermostable enzyme. So again, this is the same picture that I'm going to tell you, or this picture I will explain you here only. Like this is, as I've told, this is a PCR machine. This is the template DNA, the DNA that need to be amplified. This is the two primers that's going and going to bind to this template DNA. This is the DNTPs, that is nucleotides. I told you DNA polymerase will go and add nucleotides, but we have to give it nucleotides so that it can add to that growing DNA strand. So this is the DNTP nucleotides. This is the Taq polymerase, that is DNA polymerase, Taq polymerase, which is thermostable enzyme. This is the buffer which we need, and this is the PCR tube. So for this step we put this tube inside this PCR machine, and it is very specific tube because it is very small in size. So this is called PCR tube. So these are the PCR components which are needed to amplify our gene of interest. Let's go to next slide. So as I told, we need nucleotides also, because this is only nucleotides that DNA polymerase is going to add to our growing and extending DNA. Now, what are the steps used in PCR? So in PCR, we have this 3 steps. First is the denaturation. As the name suggests, denaturation. So the double stranded DNA is heated at high temperature. That is, even at it is approximate to 94 degrees Celsius. So at this high temperature, the double stranded DNA separates into 2 individual strands. Then now we need to cool down the temperature, means bring that temperature to approximately 50 to 60 degrees Celsius, so that our primers that are small chemically synthesized oligonucleotides that are complementary to our DNA template, so that primer can go and bind. What can the other name for bind is anneal. That's why its name is annealing. Can bind to their complementary sequence on the single stranded DNA templates that we got after this denaturation step. So it is very easy to learn this name of these steps of PCR, because this name is itself defining the what is the process or what is the procedure going on that step. So this is the first 2 step. Third step is extension. So what happened? First we have denatured our double stranded DNA, make it into single strand DNA. Now both the primers will go and bind to its complementary sequence in that DNA. Now what will happen? The temperature is raised to the optimum temperature for our DNA polymerase, that is Taq polymerase, that is 72 degrees Celsius. And now that the polymerase going to add nucleotides to that primer and synthesize a new strand of DNA which is complementary to that template. Now let me explain it with figure so that you can understand it better. So so these are the 3 basic steps of conventional PCR. So usually what happens? See, this is a 5' to 3', and here it's again 5'. It is double stranded DNA template. Now it got denatured and it became single strand. Now what will happen in second step, that is annealing? Primers will come, which has been shown in this orange and red thing, will come and bind it to the complementary part of it. Now after binding, this DNA polymerase will come at the same, whose optimum temperature is 72 degrees Celsius, and will start adding nucleotides. Starting nucleotides and going to add nucleotides to the primer and extend our new strand of DNA. And now you can see from 1 we got this 2 same DNA template DNAs. Okay, so what is the outcome? If the process is repeated many times, the segment of DNA can be amplified to approximately 1 billion copies in about 30 cycles. It means if we will repeat this PCR cycle for 30 cycles, we will get approximately billion of copies. It is a huge number. This is the same diagram which you guys have seen from last few slides, in which here is the PCR components, that is template DNA, 2 primers, DNTPs, Taq polymerase, mix buffer, PCR tube, and this is a PCR machine, where this is the 3 basic step of conventional PCR, which includes denaturation, annealing and extension. So what is the purpose of this? As I told, its purpose is to synthesize multiple copies, amplification of a specific gene or a DNA sequence outside the living organism in laboratory. We separate this by using gel electrophoresis, which we have talked in last class. So, what are the requirements? What are the requirements? So, as I have told, this DNA template, that DNA that needs to be amplified, primers, which are the two sets of chemically synthesized small oligonucleotides which are complementary to our DNA, to the regions of the DNA polymerase, which is a thermostable enzyme, typically Taq polymerase, isolated from the bacterium Thermus aquaticus. It's remain active at the high temperature, and the nucleotides which DNA polymerase is going to add to the primer and synthesize the new DNA strands. What are the three steps? We are repeating it again. First, the denaturation, that double stranded DNA is going to be heated at such a high temperature that it separates into the two strands. The next step involves annealing. Now, mixture has been cooled down to 50 to 60 degrees Celsius from 92 degrees Celsius, and now we will allow the primers to bind anneal to their complementary sequence on the single stranded DNA templates. Now, when this primers went when this primer go and bind to the complementary region, after that the temperature is again raised to the optimal temperature of DNA polymerase, that is 72 degrees Celsius, and this polymerase add nucleotides to the primer, and like this synthesize a new strand of DNA which is complementary to the template. What will be the outcome? If the process is repeated many times, as I told, the segment of DNA will get multiplied, amplified, and approximately we can get billion of copies in about only 30 cycles. So what is the formula by which we can calculate that how many of DNA copies we gonna get after amplification? So here is the formula, that is n equals to n naught into 1 plus e power n. Here n denotes at last the total number of the DNA which we are gonna get, number of the DNA. This is the initial number of the DNA, whereas e represent the amplification efficiency. What is the efficiency of that PCR machine? And n represents the number of cycles. As I said, where n represents the final number of DNA copies that we're gonna get after PCR, and not represent the initial number of the DNA copies. E represent the amplification efficiency, which ranges from 0 to 1, with 1 being 100% efficient and 0 being 0% efficient. N represents the number of the PCR cycle. So, if we see that if that PCR machine is very ideal and theoretical and its efficiency is totally 100%, then we say that E is 1 and each cycle doubles the DNA amount. So now, as you guys can see, what was the formula there? N equals to n naught into 1 plus E ki power n. Now 1 plus E, if E is 1, then 1 plus 1 is 2. So then it is like n equals to n naught 2 ki power n. 2 raised to power n. So after we got our amplified DNA, now what we will do? The source of DNA, the gene of interest we do. Now, as we have cut this gene of interest and vector DNA with the same restriction enzyme. This will generate the complementary sticky ends on both the DNA fragments, as we have seen in the last few slides, like this. That this is the, for example, this is our gene of interest, and this is the vector DNA. We got this sticky ends, and it going to ligate together using DNA ligase. So the cut DNA fragments, that is gene of interest and vector, are mixed together. The enzyme DNA ligase is added to the mixture, and like this we will get our recombinant DNA as shown in the figure. That how these two sticky ends that we have generated after we use this restriction endonucleases, we got our recombinant DNA. This DNA ligase act as a molecular glue. How it acts as a molecular glue? It forms the phosphodiester bond between the DNA fragments and the vector DNA, and it results in the formation of recombinant DNA molecule. This is the same figure. Now, after we got this recombinant DNA, now it's time for insertion of this recombinant DNA into the host cell, host organism. What is the process? Now, this recombinant DNA is introduced into the competent recipient host cell. We have talked about what is competent host cell in the last class, the cell which is suitable to take up this recombinant DNA. By how? Like if its cell membrane and cell is made in such a way that it can receive this recombinant DNA, it became a good competent host cell. The most commonly used host is E. coli, and this process in which this transformation take place is this process in which this recombinant DNA go into the competent host cell is called transformation. So what are the steps in transformation? We make these our cells competent, that is receptive, using various methods, chemical, physical and biological, which we have talked in last class. Then we add our recombinant DNA to the cell. We give it some heat shock at 42 degrees Celsius or any electrical pulse. As I told, that first we have to make our cell competent, then we have to add DNA to the cells, and then we give this heat shock 42 degrees Celsius for some time, so that they can make, so this cell membrane can make some transient port, and that port allow DNA for this recombinant DNA to get inside the host. This is the pictorial representation, that how by using injection or this gun gene method we can transform our host and make a transgene. Now what are the steps in transformation? First we need recovery in growth medium, selection of transformed cells, verification of successful transformation. We made our host a transformed cells, okay, but now we have to grow it into again media, culture it. Now we have to select a transformed cell, then we have to see that, using selection marker, that how our host or our cell got transformed. So these are the steps we use after transformation. What is the outcome? This I will. If the recombinant DNA carries a gene for antibiotic resistance, example ampicillin resistance, the host cell become transformed to ampicillin resistant cell. So what is saying that if the recombinant DNA, like for my recombinant DNA, it is a gene which is making a host, or it is encoding a protein which will make the host the antibiotic resistance, that is the ampicillin resistance, the host cell to which this recombinant DNA has got into that host cell will become transformed into ampicillin resistant cell. It will follow this phenotype. This trait, that it will become ampicillin resistant. How we will select our, that okay, this is our recombinant this is the host, or this is our, in this host, this recombinant DNA got inserted into this host cell. How we can select is that the transformed cells are grown on an ampicillin containing agar plate. Only the transformed cell with the recombinant DNA will grow. The untransformed recipient cell will die, and the ampicillin resistant gene in this case is called selectable marker. As I told you guys, that the recombinant DNA which we have transferred into the host cell, its function is to make this cell ampicillin resistance. So how we can check that the transformed cells are made into ampicillin resistance? We will grow our transformed cell on a agar plate who has this ampicillin. Okay, so now what will happen? Only the transformed cell who has this recombinant DNA, means who has the power of this resistance towards this ampicillin, will grow. The untransformed recipient cell who don't have this resistance against this ampicillin going to die. And here this ampicillin resistance gene is called the selectable marker. The selectable marker we have also studied in detail in last classes. Now we have did everything. We have make a recombinant DNA, we have got inserted into host, then we have selected our even transformed cells. Now what we will do? Now we will obtain the foreign gene product. So when alien DNA is inserted into cloning vector and transformed to the cell, it multiplies. Main goal is to produce desired proteins through recombinant DNA expression. Expression steps are clone gene of interest, optimize condition, induce protein production. So what actually this whole slide summarize about is that when we will take our when we are taking our foreign DNA and insert into this cloning vector, and now this whole recombinant DNA will go and transfer to the cell, it will multiply. So when it will going to multiply, so our product, our gene product, our foreign gene product is going to multiply, and it will produce desired proteins through recombinant DNA expression. And what are these expression steps? First, it clones the gene of interest, it optimizes conditions, and it induces the protein production. After this, we got our foreign protein. Now what we need to do, the biggest step, that we need to commercialize it, to make it to a large scale production so that it can be useful to the mankind. So we have to commercialize it to make it into the large scale production. We should meet market demands. We cannot make some recombinant protein and which is not even useful to the human. So we have to make something which is meeting the market demands and some industrial applications. So what is the process of obtaining the foreign gene product? Large scale production is needed for recombinant protein. Protein produced when a gene is expressed in a different host organism. Production process: grow cells in laboratory, extract desired protein, purify using separation technique. So it's again telling the same thing, that is the recombinant protein. This is a protein produced when a gene is expressed in a different host organism, means gene is taken from one source and host is another source. Then this protein is produced when this gene is expressed in the different host organism. This is what it's gonna tell. It's mean like it's from this gene and this host are from different sources. What are its production processes? That first we have to grow cells in laboratory, then we have to extract this desired protein, and we have to purify this protein. We can't take this protein in, it's if having some contamination or something, we have some separation techniques, purifying techniques before use this purified techniques before this protein, recombinant protein, makes to the market. Now how will we do this large scale production? We use bioreactors. I have all told about this bioreactors in the first class, but I will give a brief again. That bioreactors are the large vessels. It can vary, its capacity can vary up to 100 to 1000 liters. They biologically convert raw materials into specific products, and it provides an optimum conditions. How it provides optimum conditions? It can set optimum temperature, pressure, pH, everything, substrate, everything, so that we can get a foreign gene product. They use microbial, plant, animal, or human cells for conversion. What are its advantages? As I have told, that it provides optimal condition for achieving the desired products. What are the controlled growth conditions which they can provide us is temperature, pH, substrate, salts, vitamin, and oxygen as well. These are the conditions, these are the optimal condition which they can provide, and we can achieve by this our desired products. What are the types of bioreactors? So in NCERT, it's mentioned that we have. So first, this simple state tank bioreactor, it is usually cylindrical with curved base so that it can facilitate the mixing properly. It contains a stirrer that ensures even mixing and oxygen availability. Here in this diagram, you guys can see this is the simple state tank bioreactor. It is cylindrical. It ends are curved. Okay, it has a stirrer. The stirrer rotates so that oxygen mixing happens properly, and to every cell there is an availability to oxygen. Second type is sparge stirred tank bioreactor. In this, sterile air is bubble sparged through the reactor, increase the surface area for oxygen transfer. I will explain it in diagram. So like here, what happened from this sparger, as you guys can see, this is a T-shaped sparger. From this sterile air, means clean air, decontaminated air, is bubble sparged through the reactor so that oxygen is reached to every point, and it increases the surface area for oxygen transfer. This is something, 2 points that you guys need to remember. Then after all this, what are the last step? That is the downstream processing, which is a series of processes following the biosynthetic stage, that is fermentation in the bioreactor. After that stage, we have to do this downstream processing. The product is separated and purified before being marketed as a finished product. In short, in this step, we will separate and purify our product so that it can be made suitable for the market use. So, what are the steps involved in downstream processing? Separation, as I told, the product is separated from the culture media and cell debris, everything, and then it's been purify also. So these are the main downstream processing. After this fermentation and all those things in bioreactor, we got products plus impurity, in which we have culture medium, cell debris, and many more things. First we separate it, then we purify it, then we formulate it. So formulation means like we will see like how much amount of what is needed in it. Purification, the product is purified to remove impurity and other byproducts. Quality control and clinical trials. After purification, that is after all those steps, last is to check the quality. What is the quality? And in case of drugs, it can even go into clinical trials. What is clinical trials? Clinical trials is using that, for example, if we have make a recombinant drug, so that drug we can use if it pass every step of purification. If it works good in vitro, in vivo, everything, then we can take it to other step, that is clinical trial, to ensure its safety and efficacy. What is significance? Downstreaming processing and quality control vary depending on the product. These steps are critical for determining the final value and usability of the biotechnologist product. So as I told, these steps are important because by this steps we can see what is the quality of our product and all those things. And is it feasible to go to market or all. So now what is the summary? In summary, we have done, like, first we what did at how we isolated our DNA, how we cut that DNA and fragments separate using gel electrophoresis. Then we did PCR, that is, mm, then we did PCR, that is, amplify the specific DNA segments through thermal cycling and DNA polymerase. Then we ligate our foreign DNA and vector using DNA ligase. Then we have inserted our recombinant DNA into bacteria or cell, which we call host. Then we make sure then what happened, that host produce desired protein by expressing our gene. Then this downstreaming process happen. In this down process streaming, what we do, we isolate the product and purify it. So what are the keynotes? That PCR was invented by Carey Mullis in 1983, for which he has won Nobel Prize in Chemistry in 1993. Then the first GMO, that is genetically modified organism, whose definition and things we have talked in first class, was antibiotic resistant tobacco, which was in 1983. Then our human genome contains approximately 20,000 genes. The discovery of DNA ligase happens in 1967, which has revolutionized this genetic research. Then this DNA ligase used in treating hemorrhoids and even various veins. Let's talk about this NCERT based questions. Explain the process of DNA ligation and it's important. We know what will be the solution, that DNA ligation is the joining of DNA fragments. It is performed by DNA ligase enzyme. It creates the phosphodiester bond between 3 hydroxyl end of one DNA fragment and 5 phosphate end of another fragment. It requires ATP as energy source. It is essential for creating recombinant DNA molecules, joining or inserting this foreign DNA into with vector DNA and closing circular DNA molecules. The second question says, explain the role of PCR in recombinant DNA technology. We know that polymerase chain reaction, it amplifies the specific DNA sequences. Components needed are template DNA, primers, nucleotides, DNTPs, what we call DNA polymerase, that is stack polymerase. What is application? It's help in gene cloning, DNA sequencing, diagnostic tools, mutation detection, and even gene expression analysis. These are the interesting questions that you guys gonna solve yourself. What is purpose of using selectable markers in recombinant DNA experiments? How does polymerase chain reaction amplify specific DNA sequence? Why it is necessary to identify recombinant cell after the transformation cells process? And how does a DNA ligase facilitate the joining of DNA fragments during the creation of recombinant DNA? Thank you. And we will solve DPP questions. So let's start. First question says, during restriction enzyme digestion, why does DNA move towards end node in agarose gel electrophoresis? Let's see the options. First option says DNA is positively charged and is attracted to negative electrode. No. DNA is negatively charged and is attracted to the positive electrode. Yes. Whereas the second and third option says DNA is neutral. No. And fourth says DNA binds to the agarose matrix. No. Next question says, which enzyme is responsible for joining the gene of interest with the vector DNA during recombinant DNA formation? Like, what is the enzyme by which we can join this DNA ligase? Oh, sorry, I have told the answer, sorry. We can join this gene of interest and vector DNA during recombinant DNA formation. So the answer which I have told by mistake is ligase. In PCR, why is thermostable DNA polymerase, that is, the Taq polymerase, is essential? First option says it can synthesize DNA without a primer. No. It remains active even at high temperature used for denaturation. Yeah, it's true. It prevents the formation of secondary structure in DNA. No. It works only at low temperature. No. It works at temperature. What is the purpose of primer in PCR? First, to denature the DNA template? No. To provide a starting point for DNA polymerase to extend and amplify the target sequence? Yes. The third and fourth options are to cut the DNA? No. It is what restriction endonuclease do. And fourth, to ligate DNA fragments? No. Now, last question. How many copies of the target DNA are theoretically produced after 30 cycle of PCR, assuming perfect amplification? So, as I have told you, like if there is perfect amplification, that is okay, 1 billion, good. So, as you guys have already given the answer, but let me explain also that we know that if there is perfect PCR, then the formula will going to be n equals to n naught 2 raised to the power n, where n is the number of the cycle. So if you guys will see, if assume that first that n naught is 1, that initially we have only one DNA, so it would n equals to be 2 in 2 raised to the power n, where n is 30. So if you will calculate 2 raised to power 30, the answer will come out to be this. So this is the answer. Let's save it and submit. We got all the answer correct. Thank you